Hi, in these notes today, we're talking about DNA replication. So, the structure of DNA has some inherent qualities that allow it to make copies of itself or replicate pretty easily. And that concept lies in base pairing, that A always binds with T and C always binds with G. We say that each strand, you can almost cut it down the middle and it's complementary. So in your note sheet that I provided you, you had kind of these pieces to fill in, and this is a model or a representation of DNA replication. So there's essentially kind of two sides. We see there's an A and a B, and in terms of our first steps, we have DNA helicase, unwinding and unzipping the DNA molecule. Remember that DNA's structure is a double helix, and DNA helicase is an enzyme that unwinds and unzips the DNA molecule, the double helix. DNA polymerase then comes in and binds free floating nucleotides. They're kind of represented by these puzzly looking pieces here. They look like blocks fitting in. And DNA polymerase, another enzyme, comes in and binds these free floating nucleotides to the original or template strand of DNA. So just some clarification here. I'm going to put it in red because I think that's going to show up really, really nice. Out here, this is the original strand of DNA, and it extends all the way up here. And then we have our new complementary, I'm going to use blue, strand of DNA coming in here and binding. So the original is in red, the complementary or new strand of DNA is in blue. Okay. And this process is happening pretty quickly. It's really remarkable how quickly all of this occurs in a living thing, at least, in my opinion. So moving forward, we have what's called the leading strand, and the leading strand is A, pictured here. The leading strand's going to add those free-floating nucleotides continuously. Okay, very smoothly, as you can see highlighted in yellow. The lagging strand is a little bit different. The lagging strand adds the free-floating nucleotides in little fragments called Okazaki fragments, and they're highlighted there. And these fragments eventually get glued together by a molecule called DNA ligase. This is yet another enzyme. So this is our summary here of what's happening during the replication or the DNA copying process. There is a lot of detail there, but we can summarize it by using a couple of different models. Here we have the original strand of DNA in like a turquoise blue, and we also call that the parent or the parental strand. And then in like a pink and red, the new or daughter strands are forming. Here's another example. These are all models or representations of the replication process. The DNA molecules opening up, free-floating nucleotides are coming in and binding, and we can see that the two resulting strands are identical to the original or parent strand. Here's a summary in words, exactly what I just kind of explained. DNA helicase comes in and unwinds and opens up the DNA molecule, and each of those parent strands serves as a template for those complementary bases that come in, and it's DNA polymerase that adds in those complementary bases. Also, interestingly, DNA polymerase proofreads and makes sure there is no mistakes made. So we say that replication is semi-conservative in that the original strand of DNA is actually used or conserved in the new strands that are created. And these complementary base pairing rules allow the resulting two DNA strands to be identical. And I just thought these are some fun little facts. DNA replication happens at about 50 nucleotides added per second, so it's really, really, really quick. The process takes a few hours, and there aren't really many errors, which is also, I think, pretty neat. I also posted in the presentation some links that you can follow if you'd like to see additional models, animations, or activities. Thanks again.